while you're expecting, so.
All right. Yeah. Well, it's 4:15, so typically a few more people will come on, and but we can go ahead and get started. And welcome back, everybody. I can't see anyone's face. Let me see if I can rearrange this. There we go. I hope that you all had a gentle easing back into class this week, and hopefully got to rejuvenate a little bit last week. But thanks for joining. Before we get rolling, I just wanted to tell you about an opportunity on March 8th from 4:30 to 5:30. We're going to have the director of Seismic's gift program talk to you guys about the fellowship opportunities. And I know that Stan has done that twice, and he, loved it. Great experience. Janet Standevan has done that as well. Have you, Cynthia? Or you were going to, and then COVID.
I've done it 4 times, and every time has been phenomenal.
Yeah, and I don't. Am I wrong on this? Have those opportunities led to further opportunities for you?
Absolutely.
Yeah.
And I know people that have done it every year for almost 10 years because a professor or whoever an employer will request them. So they're doing the same job. They're going and doing research every year, in the same facility, which is awesome.
Yeah.
If that's what you want to do. If you want to have a completely different, experience, you can do that as well.
Yeah. So typically it's around 7 weeks. It's paid. And it's interactive. So and last summer they actually did a few that were virtual, but knock on wood, hopefully this summer it can be in person, because that would be the best. But anyway, so I'll stick those details in the chat, and just if you want to put a hold in your calendar for March 8th at 4:30 to 5:30, so it's another Monday. So hopefully this has been okay for you guys, this time frame. That's that. And then we'll kick it off with Emily Gleason from Mini PCR.
Hi guys, I'm Emily. I'm a part of the Mini PCR Bio's curriculum team, and I'm filling in for Katie Martin, who's the excellent Genes in Space lead. So normally she'd be leading you through this workshop, but she is on leave for the next few months. Luckily, I was in Katie's role prior to joining the curriculum team, so I know a little bit about Genes in Space, and hopefully can answer any questions you might have today. I put this first slide up just to make sure you guys have everything available to you. We're going to be doing a lot of different things today, and it would be good if you guys have it all. So if you want to just take a moment and check the list. Leave the XMN1, the restriction enzyme, let's leave that in the freezer for now, because we don't need it right away. But it's a restriction enzyme. It should be in 4 PCR tubes, and it would have been in your freezer bag. We don't need it right now, but make sure you know where it is.
What is it labeled?
3. That I don't know.
I don't know that it was labeled, or it might have X's on it, but it was in the teeny tiny PCR tubes in a strip of 4.
This is an MPX,
Maybe AX, BX.
Yeah, AX, BX, CX, DX.
Oh yeah. Oh, it's already labeled for you. You guys don't even need your marker then.
I just put it back in the freezer.
Is everybody, any other questions on any of this other list? It's a lot of stuff, so I just wanted to make sure we had it before we got started.
Ooh.
All right, I am going to move on. So welcome. This is it's a part 2 of the Genes in Space workshop. You started with Bruce last week, and he covered PCR and gave you guys an introduction to Genes in Space. So today, like I mentioned, we have a lot to do. I'm going to briefly review what you did last session, because I know it's been 2 weeks and there's been a lot of turkey consumed in between, hopefully. So we want to just make sure you remember what the lab is about and where we left off. And then we're going to do a lot of hands-on stuff. We're going to pour a gel together. We are going to do a restriction enzyme digest. I don't know why I put that all in caps. And we are going to perform gel electrophoresis and get the results of your experiment. And while we're waiting for the results to come to run, we'll talk a little bit more about the Genes in Space contest in 2021, and then wrap up and answer any questions you might have about the whole suite of things that Bruce and I have covered over the last 2 weeks. Sound good? All right.
Ooh.
Last session. So there was, as you look at you guys, there was a crisis aboard the International Space Station. The astronauts are getting sick from foodborne illness, and we wanted to know, was it the space sushi or the space burgers that were contaminated with pathogenic E. coli? So last week, you guys were looking, used PCR to take from a complex sample that you collected from your food, or not you, but hypothetical scientists in this scenario. You amplified the region of interest, the region of that bacterial DNA that differs between the pathogenic strain, or a region that differs between the pathogenic strain and the normal strain that we find in most places, using PCR and micropipetting. And today, and as you remember, PCR is this technique where we're able to identify a region of interest in our DNA, find it in a complex sample, and make billions of copies in order to be able to analyze it. And that's what we're going to do today. So we're going to go on and perform the restriction enzyme digestion, which will let us know that you will help us differentiate between that DNA from pathogenic strain and DNA from a non-pathogenic strain. And then we're going to look at that, separate those pieces using gel electrophoresis, and use that to interpret those results and get our results. Any questions before I move on? All right. So now we're going to jump into the hands-on piece. I have never done this over Zoom before. I usually walk people through it. We're going to see how this goes. We're going to pour a gel. So this is what you need of that laundry list of things that you needed. You're going to need that little tablet. A gel green tablet is a beige gel. It is in your room temperature baggie. And then you're also going to need a white casting platform and a clear gel tray and a white comb from your blue gels. So if you guys want to locate that stuff. And then we're also going to need, this is the microwaving part. So we're going to need, or your hot plate part, we're going to need your heat source, your graduated cylinder, some distilled water, and a microwave safe beaker of some kind. Everybody good?
For those that aren't super familiar with the blue gel, in the baggie that the blue gel came in, there was a white little
Yeah, I got
tray.
a picture.
There it is. There's a picture.
Yeah.
Well, and the gel tray was in the blue gel itself. That we pulled out last week.
Yep. Does somebody want to hold it up? I actually didn't realize today that I do not have a blue gel at home because I don't do that at my house on a normal basis. Yep, you got it. So the combs are going to be on the underside. Yep, and there should be a clear casting tray that fits right in there. There's one slot on one side of the white casting tray and two slots on the other side, and you'll see matching bumps essentially on that clear casting tray that should fit right in one direction. All right, so what we're gonna wanna do is you're gonna take that heat-proof flask that you have, you're gonna pop that, and you're just like popping an aspirin out of those blister packs or whatever, you're gonna pop that gel green tab into your flask and add 20 mils of distilled water. And then swirl it around a little bit and let it sort of dissolve.
So this is the gel green tab, like it looks like this?
Yep, that's it.
And then
So I have one that's white. Is that the same thing?
Yeah, it's beigeish.
Okay.
It should look a little red when you dissolve it in the water. And it should be sort of horizontal. It should not be a circle.
Yeah, gotcha.
It looks like a vitamin.
Don't eat it, please.
Yeah, it's not a vitamin.
It is not a vitamin.
How much distilled water goes in this?
20 mils.
All right.
If you want to, I think there's some evaporation. I see a lot of evaporation. So if you want to pour like a little over 20 mils, it doesn't have to be perfectly precise.
Gotcha.
And then when you have that ready, it does not look like, it's kind of hard to say. Can somebody yell when they have theirs ready? So, oh, that looks good. Sheena, do you have a larger container? Is that, because it's going to boil. Yeah, that's better. It's going to boil over.
Randy's swirling.
All right, Brandy looks good.
Stan's getting there.
All right.
All right, and then if you're using a microwave, you're gonna wanna pop hold your container into the microwave. I like to do it for about 30 seconds at a time and mix it between, just so it doesn't boil over and so you don't have as much evaporation. And as you're mixing it, you're gonna hold it up to the light and you're gonna look and see if everything's fully dissolved or if you can see little crystals floating in there. If you can see little crystals, you gotta keep microwaving. If it all looks uniform, it's ready to go. This is so exciting. I've really never poured a gel in Zoom.
Nice.
And if you're in doubt at all about whether or not it's fully dissolved, you can go ahead and give it another 10 seconds. And if it looks like you're getting a lot of evaporation from your water, you can always add just a tiny bit more distilled water. Pouring gels is not a completely precise science. All right, I'm gonna just move on to the next slide in case anybody's there. We're gonna wanna make sure that our casting tray is in our casting platform. And at the top of that gel, again, the combs fit right in so that the highest up on the gel or on the casting platform that you can put it, you're gonna put the comb using the larger, with the larger side down. So the opposite of what my picture is showing. So that those little teeth, you want the larger ones to be facing down at the top of your gel. And then you're gonna gently pour your completely melted agarose mixture in there.
So do we want the nine tooth? Is that what you said, or
The nine tooth, yes.
Okay.
So the bigger one, the ones with the bigger piece, you want that to be down.
Gotcha. And we only want to pour it in the first tray. Is that correct?
You only need one gel. We only made enough for one gel. So, Sheena, you're muted.
Oh, I just was saying I already poured it all in one.
Perfect. That's what we're doing.
We only used one of the combs.
Yeah, and you only need one comb.
Sweet. All right, cool.
All right. Can I get, if people, it looks like some people are still pouring.
Good.
If other people want to check in as you're, I see Brandy is good.
Stan's good.
Stan's good. Awesome.
Yeah, I'm back. I had to go out the room to get it.
All right, so you want your casting tray, the clear casting tray to be in your casting platform, and you want the 9-well comb to be down,
Got that.
the bigger one, and then just pour it right in there. And it looks like we're mostly all set. Is that, yell if you want me to pause. I don't want to rush you, but I also don't want you guys to be sitting there bored and waiting for me. All right. So while that gel is solidifying, we're going to go ahead and do our restriction enzyme digestion. So how many, just a show of hands, how many of you guys teach restriction enzymes in your classes? Awesome. So this is going to mostly be review for some of you. That's good. So how this works, we amplified a sequence that's found in both the pathogenic and non-pathogenic E. coli. The sequences of their genomes are really, really similar, but there's a few small differences, and we've amplified a piece that contains one of these differences. And the difference is a restriction, or it's a single nucleotide polymorphism, which means that it's just one base pair different between the two sequences, and that creates a unique restriction enzyme site in the pathogenic E. coli that's not found in the non-pathogenic strain. So we're going to use this restriction enzyme XMN1 to cut or to expose it to both our DNA, and if there's the pathogenic E. coli sequence, it will cut, and if there's a non-pathogenic E. coli sequence, it will not cut. And just as you guys may know, restriction enzymes are at work as essentially molecular scissors. They're naturally found in bacteria, and they can cut DNA at really specific sites called restriction sites. In bacteria, this is a defense mechanism against viral infection. Bacteria are under a lot of pressure from viruses that infect them, and they've developed evolved all of these tools. And we've taken it and used it in the lab, and there's literally hundreds of restriction enzymes that each target a unique sequence between 4 and 14 base pairs long. So here's some examples. None of these are the XMN1 we're going to use today, but these are some really common restriction enzyme sites. Again, to walk you through the experimental plan, we use PCR to amplify this PhilC gene from E. coli in the pathogenic strain. And so that's going to be a 400 base pair piece. In the pathogenic strain, there's an XMN1 site. So when exposed to XMN1 enzyme, this will be cut into 2 pieces of 150 base pairs and 250 base pairs, while the non-pathogenic strain will remain uncut and 450 base pairs in length. And then we're going to use that gel you just poured to separate these DNA strands by size. And we'll be able to tell, is there 1 band or 2 bands and how big they are. So for the restriction enzyme step, now we need that XMN1 enzyme. So it's okay to take that out of your freezer now. And you need your PCR reactions from last week, as well as a micropipette, a tube rack to hold your tubes, and your mini PCR thermocycler, along with a laptop to program it. We'll pause while everybody grabs their XMN1. All right, are people's PCR reactions frozen, or were they stored in the fridge?
Mine were in the fridge.
In the fridge?
Frozen.
Frozen. If they're frozen, you want to hold them in your hand for a second and thaw them a bit, because we're going to start pipetting them. What we're going to do is we're going to take 15 microliters of PCR product from each one of your tubes and add it to one of your restriction enzyme tubes. So A should go in AX, and it sounds like they've been pre-labeled for you. B should go in BX, P should go in PX, and NP should go in NPX. And there's 1 microliter of restriction enzyme in that tube. You also may want to shake it down. The restriction enzyme, may have come up to the top. You want to make sure everything, once you've capped the tubes, you've added it, then shake it or tap it on the table to make sure all the liquid is at the bottom, because we want the restriction enzyme and the PCR product to come together. And 1 microliter of XMN1 is not going to look like much. So it may be hard to see.
AXBX. Okay, some of these are rubbed off.
Yeah, if your label's rubbed off, I think it's the same thing in each one of those tubes. So just label it so that you know what it is now.
We're just taking 15 microliters and putting them into the restriction enzyme.
Right, yeah, add it right into the restriction enzyme, and then make sure everything is at the bottom of your tube. If you happen to have a centrifuge, that's the safest way to do it. But if you don't, you can flick your tubes to shake everything down, or tap them on the table to sort of gently bring them down. I happen to prefer the flick method, but I know a lot of people like the tapping. Both will work.
I got a question.
Yes.
So where do I find the AX, BX, PX, and MPX?
So that's the XMN1 enzyme. You should have gotten it in 4 small PCR tubes that were in your freezer.
And our control PCR tubes with the green has the green in it.
Yes, and hang on to the rest of your PCR reaction, because we're going to run that on the gel as well as the digested PCR reactions.
So we're going ahead and adding, though, our PCR product to the restriction enzyme.
So there's 30 microliters of PCR product. You're going to add 15 to the restriction
Oh, I got you.
enzyme.
Only 15.
And then you'll have the other half available-ish
Gotcha. That's what's going to be.
to load that on the gel.
Gotcha. Perfect.
Brandy, did you find them?
No.
Okay.
I'm looking. I wonder.
Yeah, there should have been a freezer bag that had them in it.
Yeah, I've got. I don't have.
Is that a fridge bag or a freezer bag?
This freezer.
Ew.
I'm wondering if I got them mixed up with what we had to do last week.
It could be. Last week.
Oh no.
Yeah, there should have been 4 PCR tubes with the XMN1 enzyme.
No, I broke those off. I'm looking.
Okay. I see lots of pipetting going on. As you're wrapping up your pipette, does anyone need, if I move on to the next slide. So we're gonna, restriction enzymes naturally occur in bacteria and function best at 37. So we're gonna incubate them at 37 using your mini PCR as a heat block. This is pretty easy to do. So once you're finished pipetting, you're gonna wanna put them in your mini PCR, close the lid, and then open up your program, set a heat block program. So that'll be like a plus on the top, and then you'll select heat block and set it for 37 degrees C for 15 minutes. Not trying to rush you, just want to make sure everybody has the instructions if they have completed that step.
Okay. So we got to amplify again here. Okay.
No, we're not going to amplify again.
What are we doing? Just getting
We're
it-
incubating at 37, and that makes the restriction enzyme, it's just sort of its optimal operating temperature. So it will cut when it finds its recognition sequence and not cut if it doesn't. Yes, I can go back one screen. Oh, that's forward. Hang on.
I really love these little bags y'all sent, to put everything in. That's super cool. I want this for everything I have. Okay. Now I was doing this with my phone before.
You have a Mini 16?
Yeah. So I guess I'm going to have to do that again.
Yep. Or you can hook it up to your laptop with a USB cord. But
Yeah, I've got a Chromebook and it won't work.
Oh, yeah, that is true. I don't know. I like programming my PCR machine with my phone because it just feels very 21st century. That was not available when I was in grad school, so I think it's fun. But
I thought it worked really well.
yeah, when we used to work in an office back before COVID, I used to be able to program the PC, like sit at my desk and program my PCR machine in the next room, which I was super excited about.
How do you set this thing to a heat block?
So when you hit the plus program, it'll give a choice like PCR, heat block, flex. And you want to select heat block.
Okay, so once you've got all the stuff in the tubes, what do we need to do with the mini PCR machine? Sorry, I was missing my cord.
So we're going to program a heat block to 37. That's going to incubate the XMN1. It basically operates best. It will operate at room temperature, but it operates best at 37. That's body temperature. That's where these E. coli, that they're derived from.
So how do I set the device up again?
So are you using your phone or?
No, I'm hooked up to my computer.
So you want to make sure that it's on. No, don't turn it on.
Okay.
Hook it up to your computer using the USB.
I already got that.
Open your Mini PCR software.
Got it.
Hit plus for a new program.
Oh, okay. So we go back to library. All right.
Yeah, library plus for a new program, and then select heat block.
Okay.
And then you can just input what I have on the screen here, 37 for 15 minutes.
15 minutes.
15 minutes.
15 minutes. Okay. Wait.
Save and run.
Go ahead and do it now.
Yep, save and run. Yeah, if you've got it all set up, save and run, get it going.
Okay. Oh wait, do we put it in before we do save and run?
Yeah, put your tubes in your PCR machine and hit save and run and then turn it on. So we've had this issue where like sometimes the PCR machine will start running the program you put it on, and if the restriction enzyme goes up to 94 degrees, which is the first step of your PCR program, it'll get fried. So you want to just definitely make sure that you're overriding your last program with this 37-degree heat block. Does that make sense?
Doing
Emily, I
new
was gonna see if
heat
you
block
could see- I didn't have-
37 for 15
Wait, Laura,
minutes
say it again.
and save and run.
Last week when I hooked up, it worked fine and I could get in and access and change that stuff, but I'm having issue, my USB is
Because
plugged
there's no
in,
power. Oh,
but
now
I
I
can't get to that screen where I changed the temperature. It's not coming up and I'm not really sure.
So you're gonna have to input a new program. Is that what you're having trouble?
I can't even get to that first screen where I do that. Like I see the mini PCR program on my computer, but when I I don't know how to open it, I guess.
It should be a double click. Do you have an iPhone? And I don't know if you guys have Mini 16s or Mini 8s, but if you have a iPhone and a Mini 16, you can program it with your phone too. You just need to download it from the App Store.
Okay.
But yeah, regardless of whether the PCR machine's plugged in to your computer, you should be able to open the program.
Just when I don't know, I'm not very computer literate, obviously. When I open it, it doesn't bring me to that screen. It just is a list of all these different folders, like audio platforms, etc.
That may be like the download. You should have like a little M app. You didn't have a problem with it last week, though.
No.
Are you on Windows or Mac?
Windows.
Windows. So open the little window icon to the bottom left of your screen.
Uh-huh.
Find the mini PCR folder there. And then click on the one that has it's like a graphic M.
I don't know if maybe the system changed something on my settings over the break, because I see that, but I can't, it won't let me open it, so. I'm not
Oh,
really-
bummer. Do you have an iPhone?
Yeah.
Download the mini PCR app from the App Store. Megan, do they all have mini 16s? I'm not familiar with the equipment we sent you.
Two mini 8s out
Do you
there.
know-
We borrowed them. I don't remember who has them off the top of my head.
Okay. Laura, can you look at the side of your mini PCR machine and tell me if it says mini 16 or mini 8?
It's a 16.
It's in 16, perfect. So download the mini PCR app.
Okay.
And then you should be able to, and make sure Bluetooth is turned on your phone. Let me just find it on my phone just to make sure I can walk you through this. Do you have it downloaded?
Yes.
All right, so hit the M at the top of the screen.
Okay.
And then you should be able to search for your device. Make sure it's turned on. And then you want to click the Bluetooth symbol to pair it.
Okay.
And then go back, once you're paired, go back and hit the plus at the top. It's next to the little bell. And
Okay.
you should be able to set a heat block, choose heat block, and
37 for 15 minutes.
37 for 15 minutes.
Okay.
And make sure your tubes are in there and go ahead and save and run and it should be good.
Okay, thank you, sorry.
Oh, no worries. Glad you had the 16. All right, is everybody good with, I'll ask it this way. If you're not currently incubating your restriction enzyme digest, make a noise so that I know. All right, I'm gonna go ahead and move on. We're gonna talk a little bit about gel electrophoresis and then load our gels. So gel electrophoresis is a method for separating molecules. It separates based on two factors, one size and two is charge to mass ratio. But because DNA is a polymer made up of a series of monomers, it has the same charge to mass ratio. So only the size matters in this case. And what you poured earlier in the session was an agarose gel. Agarose is a long sugar molecule, essentially, that when you heat it and melt it, the sugars connect in this kind of web-like form. This is a scanning electron microscope image of what the gel would look like. And it makes all these different pores. It's a little bit like, it's really not that dissimilar from Jell-O. And so when we run an electrical current across the gel, which is what we're going to do when we're performing gel electrophoresis, the negatively charged DNA is attracted to the positive end of the electrode. So it gets pulled through the gel. But larger pieces have more trouble making it through the pores in this web-like structure, and they travel more slowly, while smaller pieces of DNA travel more quickly. And this allows us to separate the DNA by size. So we're gonna go ahead and get started setting up. I know you probably have some time left on your restriction digest, but there's a decent amount of setup for the blue gels beforehand. So we're gonna just get that going while your samples are cooking. So right now you wanna get your blue gel out. You wanna plug it in. And you wanna put the clear buffer chamber in the blue base. And then you want to make sure you have all of the stuff ready. Your XMN1 digests are still working. I understand that, but you want your 100 base pair DNA ladder, your micropipette and tips, that 1x TBE buffer that you have, and your blue gel setup.
Not have 1x right now.
They have 20x right
They
now.
have 20x. Yeah.
Ah, okay. So extra step. We're gonna need probably about 25 mils. Let's make 40. How much 1x do they have?
1 second.
Okay, there it is. So we're gonna run it in the blue gel.
We are gonna run our gels in our blue gel, yep. And the first thing we need to do, I thought you guys had 1x TBE, but you have 20x TBE, so we need to dilute it.
So they should have 2.5 milliliter tubes of it, I think, to get to 5 total.
So that's 50? Or it would make,
they should have enough to make 100 total.
100 total.
So that they have some next week too.
Okay, that was gonna say, I know we're saving the gels for next week. All right, so you're gonna use
I
the.
see Shanna holding them up.
I can't see everybody at once. I keep flipping back. So you're gonna use the, you need something that will hold 100 mils of water, and you're gonna add your 5 mils of 20x TBE and 95 mils of water, and mix them together.
Say that again.
100
All right.
mil.
So you have 5 mils of 20x TBE total, and you're going to add that to 95 mils of distilled water in a container that will hold that amount.
All right, I'm sorry, I'm walking around here in my lab. 90 milliliters of distilled water or 95?
95 milliliters of distilled water and
Okay.
5 milliliters of 20x TBE.
So if you have a 100 milliliter graduated cylinder, you can just dump in the 2 micro tubes of buffer and then fill it the rest of the way up to 100.
XTB. Where's the XTB again now?
20x TBE. That should have been in your room temperature materials. You should have two
micro tubes.
2 microtubes of it. Does somebody who has it want to hold it up for?
Microtubes. PCR tubes or just the 1.5 milliliters?
So both tubes. Each tube should have 2.5.
Show that to me again.
Brandy had hers. Can you see Brandy's? They're clear tubes. I have the lotion tube. But this size tube.
Okay. Okay. I've got one that says is TBE.
TBE,
Yeah.
That's correct.
Huh? That's what I want. The TBE.
20x TBE, yep.
So you should have 2 of those tubes.
If
Gotcha.
you don't have 2 of those, you can make 50 and you'll probably still be fine.
Yeah, I got it. We're good. All right. Okay. TBE. Okay. All right, let me say this one more time. 95 of distilled and 5 of the TBE.
That's correct.
Gotcha.
If you have your TBE made, you want to make sure your blue gel is plugged in. There's a clear buffer chamber sitting in the blue gel base. And then you can go ahead and pour a tiny amount of the 1X TBE that you just made into the clear buffer chamber. And then pop your gel. Again, the gel's going to fit only one way if you line up the holes, the slots on the side with the bumps on the side of your gel casting tray. That's it. That's a clear buffer chamber and a blue gel, Sheena. Yep, that in there and then plug it in.
Okay.
Now, what do I do with the comb?
Take the comb out. Your gel should be set by now.
Yeah, and just wiggle it back and forth.
Yep, and just pop it straight up.
Okay.
Can somebody show that so I can see what they're doing?
And so I take the gel tray, I took my comb out and I set it in the blue.
You set it in the clear buffer chamber. And I find it helpful to have a tiny amount of TBE in that buffer chamber already before you set the gel in there, because it gets rid of bubbles.
Sheena has hers. Somebody was asking to see the gel.
So Sheena, pull the comb straight up. Perfect. And then set it down in your clear buffer chamber with a little bit of buffer in it. A tiny little bit, not too much.
A splash.
A splash, yes. It keeps the bubbles from being underneath and you'll get a better picture at the end.
All right, I'm a little bit behind y'all on this.
That looks beautiful. So now Sheena and everybody who's done that, does everyone want to look at Sheena's really quickly? So she's got her gel and her buffer chamber and her blue gel. And so now when you have that, you want to slowly pour the 1X TBE until you've just barely covered the gel. The more you pour in, the slower your gel is going to run. So you need to cover the top of your gel, but no more than that.
Okay, let me go back to making the TBE buffer here. So how much are we talking about, microliters or milliliters?
Milliliters. You want 5 milliliters of the TBE to 95 milliliters of water.
Okay. I don't know that I have that much.
How much do you have? And we could.
Yeah, I have 20X right here, but I have 2 milliliters times 2.
It should be about 2.5 in each.
Really? These are 1.5s.
It's okay if it's a little bit more dilute than that. Like I said, a gel running and pouring is not a precise science. We've actually run gels with tap water. Don't tell anyone, and it still works.
Right.
Well, you do have distilled water, so that's a little bit better.
Yeah. So basically, just pour both of these TBE, these vials of TBE,
and then fill it up to 95 or to 100 mils to make your buffer.
Okay.
Does anyone want me to go back over other instructions till we get to the point where we have a gel that's ready to load?
So once I added my 95 milliliters of water, and so I just need to pour that over my gel, but don't cover my gel.
You want to just barely cover your gel. Don't like drown it. That you want to cover the surface of your gel, but no more than just barely covering it. Don't fill up the whole chamber.
Any extra you guys can put in that amber bottle for next week.
Yeah, you guys are going to run a gel again next week.
Okay. All right. So what's the concentration of TBE here that we're running?
You've taken 20X and turned it into approximately 1X.
Yep. So 1X TBE.
And you're going to use your 2% gel as the other fun concentration.
Okay. All right. So since I'm behind, let me go ahead and grab this. Pull my gel out and stick it in the blue gel.
And stick it in the blue gel. There should be only one way to do that. If you're inserting the bumps on the side of the casting tray into
Right.
the slots in the gel chamber. So you want to make sure you line those up.
Gotcha.
And before you do that, pour just a tiny, like a little splash of the TBE in the bottom of the buffer.
Okay.
To set your gel down into it. It'll keep bubbles from forming underneath the gel.
Perfect.
And it's not, it'll still work if you don't do that, but it makes it look a lot prettier because you won't have bubbles. We're going to shine a light up through our gel, and if there's bubbles, it makes it a little bit more uneven. I hear some people have some cooked
Yep, it's cooked.
samples. Excellent.
Yeah.
Are we okay to move on to the next part? So that comb you used, you've made essentially little pockets or wells in the gel. We're going to add our DNA into those wells, and you should have the pockets, the wells should be at the top. You should see a negative sign there. That's the negative electrode, and at the bottom there should be a positive electrode. And what we're going to do when we turn the gel on, and we've now covered this with a buffer, which is going to conduct electricity essentially through the gel. Jackie has a question. What's up?
Oh, okay. Miss Brown, do we take the gel out of the tray?
Yeah, pop it up out of the casting tray, and put it in your buffer chamber, and then cover it with your TBE buffer that you made.
Just out of the white tray. Leave it in the clear tray.
Yeah. Oh, sorry. Leave it in the clear tray, but take it out of the white casting platform.
All right.
Did that answer your question, Jackie?
Did it stay in that platform?
So there's a white piece that's the casting platform, and then inside of it there was a clear tray. You want to pick up the clear tray with the gel inside of it, and then move that into your buffer chamber. Good. And then this is the tricky part. We're going to load our DNA into those pockets, those wells. And like I told you, the gel's a lot like Jell-O, so it's really easy to poke a hole into it. And you want your DNA to go into the well, but you don't want it to go underneath the gel or over the top of the gel, because then it won't it won't get pulled through the gel with the electrical current. So what I find is best to do, you want to have a really steady hand for this, is just as what's shown in the picture. You want to put your tip just in the very top part of the well. That green dye that was in your PCR master mix, that is a loading stain or loading dye, and it will actually sink into the water. So you're gonna measure out 12 microliters of each of your samples, and you're going to very slowly with your finger push down on the plunger of your pipette as you're holding the tip of the pipette into the well of the gel. And I would recommend, normally you go to the second stop when you're ejecting liquid from your gel, from your pipette. You don't want to do that this time. I would recommend just going to the first stop so that you don't push any bubbles into the well and disrupt your DNA. The other thing that's helpful a lot of times, because you want to have a really steady hand, is to hold your finger or your hand underneath, your second hand underneath the tip of the pipette man, and that'll keep it even more steady. So you hold it like this, you hover it over your well, and you very gently press to the first stop and let your DNA sink to the bottom of your well. And then before releasing your plunger, you pick up your tip out of the well, because if you release your plunger while your tip is still in the well, you'll suck up what you've just pipetted in there. Does that make sense? So you're going to want to get 12 microliters. You can start with your DNA ladder. That's the 100 base pair ladder. You want to pipette 12 microliters of that. So measure that out. hover your tip into the leftmost well of your gel.
Okay. So I have two ladders. I have in a green, it says DNA ladder, and then I have another one that just says ladder. It's purplish color.
Purple is probably ours. I don't know about the.
I have 12 microliters of it.
12 microliters.
I don't have 12 microliters of it.
How much? The ladder is more concentrated. How much do you think you have?
Maybe half of that.
Load what you have.
Okay.
If you can shake down the tube and see if you can get any more down there, yeah.
And then the other one was DNA ladder. So that's not it, right?
DNA ladder might.
It's in a green tube.
Is it also purple?
Mine's orange.
No, I can't tell what color it is. It's in a green tube.
It looks blue, but it's in an orange tube. So
Megan, do you know which one's which?
You know, that's it. The orange one that has like the bluish purple liquid in it is your DNA base pair ladder that goes in lane 1.
I don't even have enough of that.
I promise you do. If you had a centrifuge in your possession, you do. I promise.
If you shake it down. I mean, otherwise,
Get as much as you can.
Get as much as you can. I can see, when I do it, I can see it with 5 microliters. You can see the ladder pretty well. So if you have more than 5, it's worth loading it.
Okay, so I must have mixed up several different experiments here. I don't know what goes with what.
They should be the same. So,
I have a DNA sample here. I've got
no DNA samples.
I've got a XMNL. I don't know what that means.
Yeah, the X was the 4 PCR tubes.
No, this isn't a microcentrifuge tube. It's XMNL or something like that. I don't know what that is. I've got one that just says ladder on it with purplish material in there. And then I have a green one that says DNA ladder on it.
Let's try the DNA ladder.
You want to try the DNA ladder? It's clear.
Right?
It's a clear.
They're both 100 base pair ladders.
Okay.
But
XMN1 is a restriction enzyme. That's correct. But in this one, you want the 100, or you want the DNA ladder.
DNA ladder.
And then we're going to load what's left of your PCR, about 12 microliters, or if you have less than that, that's okay, from your PCR reactions that we did not add restriction enzyme to. So those tubes should be labeled A, B, P, and NP. And then in the last 4 lanes, you're going to load your AX, BX, PX, and NPX, those tubes that you just digested with the XMN1 enzyme.
Okay.
Did you say if we didn't have enough in the one that said DNA ladder, can we just use the one that says ladder?
yeah. So they should both be the exact same thing, just one is for this week and one is for next week.
So maybe, but you don't want to use both of them up this week, right?
No, please do not use both this week.
Yeah, so if you don't have 12 microliters, just load what you have. As long as it's more than 5, you should be able to see it. It may be a little faint, but you should be able to see it.
And we don't need a dye for this?
It has the blue color means it has the dye in it. And similarly, the green color in the PCR and the restriction digest is a loading dye.
I don't see a color in this one at all.
In which one?
In the DNA ladder.
All right, use the
other one is purple-ish.
other one's purplish. Then you're gonna want to use the one that has dye in it.
Okay, let's do that.
Next week you'll have loading dye to add to your samples, and so if you have a clear ladder, you can add the loading dye to it.
You say these are the same ladders, though?
In
should
theory.
be, yeah.
If one's from our qPCR kit and one's from this kit, we use the same ladder.
Okay. All right. Here it is.
So I just had 5 microliters in both. Like the orange one, I can see a drop. and it's not even, I know you can't see it. So I won't have any for next week.
So technically this week, we know what the bands you're getting, like what size they should be. And like, it's not good scientific practice not to run a ladder, but you technically don't have to run a ladder this week. You should be able to tell from the experiment what size bands you have. So I don't know if it makes, Megan, if it makes more sense for people to save theirs till next week or.
I already put it in my will.
All right, well then,
there
next
you go.
week, no ladder.
Okay.
All right.
So I can pull this out of the heat block too, all these other ones, right?
If they're done cooking, yep, you can pull them out. And you're going to load your uncut PCR reactions first. That other half of the reaction that we didn't add to the restriction enzyme.
Gotcha.
So just follow the order I have on my gel here. And then you're going to add your cut reactions.
Okay. Okay, here we go. I like these gels, this Agarose, sets up really quickly.
It's nice the small amount.
Yeah.
The small pouring, the casting tray, and everything's really small. So it'll
Yeah.
run. You'll see it runs really fast too because it's nice and small.
That's awesome. So what concentration is the gel?
The gel's a 2% gel.
2%. Okay.
But you can use those tabs and just add extra. Like, so today we added 20 mils of distilled water, but you could use the same tab and add 40 mils of distilled water and make a 1% gel.
1%. Yeah, that's perfect. Where's that one from? 1, 2, 3, 4. It's the fifth. Fifth. 1, 2, 3. So our BP and NPs are shot. We won't be using those. There's nothing left, right?
There's nothing left. You can toss those tubes.
Well,
Similar with the AX, BX, PX, and NPX, we're going to load most of it here, and you can throw the tubes away.
all right, perfect. What I'd like to hear.
I live in a zoo. You might be hearing some of the zoo animal noises in my house. If you have everything loaded, Sheena, you look like you're done.
Oh,
Go ahead, put the orange cover on it, and press the power button and get it running. I don't want it to like dissolve out while you're sitting there.
2, 3, 4, 5, 6, 7.
And the orange boxy cover, because they also got y'all's prototype thing to use with the tubes the first week.
So when you push the power button, the light should turn on. If the light does not turn on, you did something wrong, and we can troubleshoot. But there should be a little blue light next to the power button that should turn on.
Mine's green. That's okay.
Yeah, as long as the light next to the power button turned on, you're good.
I see the bubbles.
Oh yeah, sometimes they're hard to see on those electrodes just because of the way it's set up. But yes, that is the way to check. Sheena, did you also get a black fold of view with your? It should be like a black piece of plastic that's folded up.
Yep, everybody had one in their bag with their blue gel.
That. So go ahead and pop that open. So yeah, put your hand inside and pop it open.
Now, I've got a question on my blue gel.
Yeah, go ahead.
So I've got my my clear rack with the gel in it. Is there supposed to be something else in it? Is because there's a lot of play. There's a lot of play in the. It doesn't fit tightly in there.
Are you able to hold it up?
Well, I mean, no, it's in the. I've got my buffer.
So there's bumps on the side of the casting platform.
Gotcha.
Or sorry, the casting tray. So there's a clear casting tray with bumps.
Clear
There's one bump on one side and 2 bumps on the other side.
You got it. It's not supposed to fit tightly though.
It should fit pretty snugly in. So there should be, the bump should go in the slot.
Not really. I mean, there's a lot of wiggle room. I've got probably 1/8 to 1/4 of an inch on either side. Is there a size? Is there versions or something on these?
No, it shouldn't be. It's all injection molded. It should all fit.
I don't know. You got me. All right. So what do we do now that they're loaded? Go ahead and put the orange
cover on, the orange cover that fits on the top of the blue base.
Gotcha.
And then push start, and you should see a light turn on. And then that piece of black plastic, the fold of view, you want to punch it open, and yes, just like Sheena is demonstrating, sit it on top of the orange cover. Make sure that your green light stays on, that your gel is running, but that blocks out the ambient light and allows you to be able to see your gel a little bit better as we're running it.
Do we need to turn on the light?
You do not need to turn on the light yet. Once it starts running in a few minutes, you can turn on the light and see if you have DNA in there. You should start to be able to see it.
Gotcha. Okay.
You don't want to leave it on though, because it'll bleach out the dye. So you can turn it on, check, turn it off again. And
So
I encourage you all.
is this just blue visible light?
Yeah, you're using blue light and and an orange filter, and the stain that we use on the gel responds to just plain blue light.
Gotcha.
And it's safe. Safer than ethidium bromide anyways. I would need it again, but it's safe. You can throw it away in your regular trash when you're done with your gel next week. And your kids don't have to worry about it too much.
Okay.
So as your gels are running, I'm gonna just flick this yell again. So you should have loaded everything, press the run button, cover it with your fold of view. And that's just to let you look at it while I'm talking. I highly encourage you to be really distracted while I'm talking and check on your gel, because you should start to see the bands begin to separate, and being able to see that in real time is fun. We don't know if we have to, you don't have to stain the gel afterwards, so you actually get to watch as the bands separate. Is it okay with everybody if I move on to Genes in Space, or are people still finishing up? I don't see anybody loading right now. Is everybody, if you're not good, yell.
I just,
All right. What?
I just need to clarify really quick.
Mhm.
So don't keep the light, turn the light off on the blue gel machine, correct?
Yeah, you don't want to leave it on because it'll bleach out the stain eventually, and your gel won't look as bright. But you're welcome. Like you can turn it on, check your gel, turn it off again, as many times as you want.
Okay, so as long as I have it plugged in, whenever I push
Oh.
the run button, it has a green light that appears. But whenever I remove my finger from the run button, the green light disappears.
The green light disappears.
Doing what it's supposed to?
No, the green light should stay on. So you want to check that you have your cover on the correct way. So the plus electrode should be on the plus part, and everything should snap down and fit really tightly.
So I figured out what my problem is. I didn't put this in the clear tray before putting in the gel.
Yeah, you want your gel in there, and you want that covered.
So I'm running it without this. Is that okay, or do I need to stop it?
Do you have buffer?
Yeah, there's buffer in just the chamber, just straight in.
That blue chamber is not waterproof.
It's not?
No.
Well, it's plastic.
It definitely has seams where that can get in.
Okay, so let me stop it then. All right. I knew something was weird. It's running just fine. Look at it.
That's incredible. I have never done that before.
I don't see how water would get through this. It's solid. It's a solid molded.
I would
the electrodes.
I would use it in the regular
Well, we are problem solving right as we speak.
Excellent.
I'm troubleshooting everything for you. Okay, we're trying to.
Brandy, did you check all your connections? Is it staying on now? Okay, good.
Okay.
I have one question before we move forward. Before you said the light, I had turned my light on when I turned the machine on. Would mine have already gotten bleached out in that time?
No, if you leave it on for like the entire time that I'm talking, you'll see a noticeable difference in how bright it is, but you'll still be able to see everything. It's fine to leave it on for a few minutes. Just don't you forget about it. Other questions? All right. I'm going to talk a little bit about the Genes in Space 2021 contest while we're waiting for our gels to run. And then we're going to predict the results and then look at them and see what we got. All right. So as Bruce told you last week, the theory behind the Genes in Space contest, or the motivating factor, essentially we're entering a new era of space exploration. We've been in low Earth orbit for the last 20 years in the International Space Station, and for many decades before that through the shuttle program. But NASA has big plans to head to the moon, to head to Mars one day. And we don't really know, how does that sort of long-term space flight affect life? Both the human body of the astronauts that are doing the traveling, as well as the plants that they might bring with them for food, or the microbes that we bring with us everywhere. And so the Genes in Space contest allows students to come, to ask these questions that we don't know the answer to, and design experiments that might test and give you the answer to some of these questions. And the platform that they have to do all this in is the ISS U.S. National Lab. Like I said, it's been continuously inhabited for the last 20 years, so the entirety of your students' lives. There's been somebody living in space, not the same people. And it's being used as essentially a platform for testing all kinds of questions. What do we need to know in order to be able to have a successful mission to the moon, a successful mission to Mars, and potentially beyond? Sorry, Levi, Control P should have been the DNA from last time. If you want your PCR reaction to load as part P for your gel. Does that make sense? Yeah, you want the PCR tube labeled P.
All right.
I'm sorry, I can't hear you. If you don't have a P, it's okay to just skip it. But the one thing about the ISS is that molecular biology techniques, like the techniques you've been using in these workshops, were not available and not tested in space until very recently. 2016 was the first time that PCR, which you guys used last week, was performed, and it was the first time that DNA sequencing has been performed on the International Space Station. And pipetting, the micropipetting that you guys have been doing, that came even later, even more recently. So your students are really able to be space biology pioneers because these tools haven't been available in space. A lot of these questions just haven't been asked and been tested. And you'll see that, as we talk about our winners, a lot of them have really pioneered space biology. From, the first PCR experiment ever done in space was designed by a 17-year-old from New York as part of the Genes in Space competition so that 2016 experiment that I referred to, last spring, about almost 2 years ago, actually, the first CRISPR-Cas9 genome editing. Again, it was a Genes in Space experiment. It was one of the more complicated molecular biology experiments that's ever been carried out in the International Space Station, and it was designed by high school students. So, when we say your students can be space biology pioneers, we really mean it. The questions they're asking, we don't have answers to, and the experiments that they designed really have pushed the field of molecular biology in space forward. So the next contest for Genes in Space is opening on January 19th of 2021. The contest is open to students in grades 7 through 12. The students can work on their own or in teams of up to 4 students, and their applications must be sponsored by an adult educator. And in some cases, that's the student's parent or another, science enthusiast in their life, but a lot of times, almost all cases, that's their teacher. And the role of the adult educators really varied. It's kind of what you guys have time for. So you can, either, just put your name on their application and say you sponsor it, or we have teachers that use this as a classroom assignment. And so they get heavily involved in guiding them through the application process, through developing their proposals. Or we have another teacher that we know that does like a Genes in Space team, and she has kids apply to be part of the Genes in Space team. And then she picks 4 of them and they work together on one single proposal to kind of keep her own workload down. The online application is going to be available on January 19th, and the contest will close April 12th of next year. So the students will have a few months to submit their applications online in order to be able to participate. And it's an ideas contest. So today we're doing a hands-on lab to get you guys, more experience with the techniques PCR and the downstream analysis, some of the downstream analysis, but the students don't need to do their experiment in order to be able to compete. It's a written proposal. It's about 1,000 words, 5 questions, and that's all they really need to do. So it's really, they're judged on the merit of their ideas and the merit of their experimental design, rather than what resources they have available to them. And just to walk you through the contest cycle, it starts, that's what we're coming up on in January. The students will write and submit a proposal, and that really starts with asking questions about what might be happening to life forms in microgravity and under other conditions only found in space. Anything from, we know that astronauts lose their sense of taste in space. Can we give them a better culinary experience by engineering plants that they would take with them into space that have stronger flavors? We know that astronauts' immune systems are suppressed in space, and that viruses can become more dangerous after exchanging DNA with each other. So does microgravity make the viruses more likely to exchange DNA, and how is your immune system going to respond to that? And once they've asked these questions, they need to design an experiment that addresses the question. And in the past, the tool that they've been given is PCR. So they design a PCR experiment that will address the question that they have about science in space. This year, we're adding 2 new tools. So this is the P51 fluorescence viewer that you guys used in the BioBits portion of this workshop a few weeks ago. and then the BioBit cell-free protein expression system that you also use. So now the students can incorporate not only PCR or not even use PCR at all, but rather rely on BioBits to produce proteins or P51 fluorescence viewer as a way of designing and developing their experiments. So these are both new in 2021, and we'll be launching a number of resources meant to support students in using these tools in their experiments going forward, as we come into the new year. And then once those proposals are in, we, a team of people here at Mini-PCR evaluate the proposals and select a number of awardees, a total of 25. So we select, the first is Constellation Award winners. These are awarded to the school in each US region with the most number of submissions. So you guys are the Southeast region in Georgia. It's just, how many students did you have participate? It's recognizing enthusiasm. We give 5 junior scientist awards to the best proposals from the 7th and 8th grade from middle school teams. We choose 10 honorable mentions and then 5 finalist teams. Each of these 25 awardees wins a set of Mini-PCR bio equipment. So that blue gel, Mini-PCR and pipette that you guys have, we send one of those to school along with recognition from the kids, medals, t-shirts, all kinds of fun stuff. The 5 finalist teams then go on to be paired with a mentor. These are usually scientists from MIT or Harvard, and they work really closely with the student over the period of about 6 to 8 weeks to take that written proposal and turn it into a presentation and that the students will present in front of a panel of judges. And in the past, that's happened in person at a special conference for people doing research on the International Space Station called the ISS R&D Conference. Last year, this past summer, that wasn't available, obviously because of the pandemic, and we moved the whole contest online. So if your students are really enthusiastic about this and want to go and watch, they can actually go and watch these finalist presentations on the Genes in Space YouTube channel, and it's more public than we've usually had that part of the contest, but that's now available to your students as a resource. They can see, what types of questions the finalists last year were asking. And then there's a panel of judges that evaluate these presentations and select a winner. And up to this point, the winning experiment has been purely an idea. It started with a student coming up with this question and developing an experiment, and then was worked on with their mentor a little bit further into this presentation, but nobody's actually done this experiment. So that's the next part of the Genes in Space cycle. Space biology camp. The students work with their mentor again and with scientists at Mini-PCR Bio and at Boeing, our collaborators at Boeing, in order to turn that experiment into something that we're able to do on the International Space Station. And oftentimes that's hands-on. Again, this year may not be because of the pandemic, but we're trying to do something virtually like we're doing here today, where this winning student will get hands-on with the experiment. And then the last part, the most exciting, we actually launched the experiment to space. For the 2019 winning team, we just launched a few weeks ago. Their experiment's currently on the International Space Station and will be performed later this week. And so again, often the students get to go to the launch. We definitely get video feed. They can watch their experiment being performed. And then we often go on to publish the results. With the student, we work on writing it up in a peer-reviewed journal and actually publishing the results. So the students get to see, the winner gets to see everything from, the idea building of putting together a scientific proposal all the way through, presenting that data to others, the full scientific process. And yeah, the experiment launched a few Sundays ago. The kids got to participate virtually. And we're performing it later this week. But I told you, that's what the winner gets. That's really cool. We only select one winning team every year. But we have, thousands of contestants, hundreds of teams submit proposals every year. So, what about them? Why participate in Genes in Space? It's not just obviously that you might become a winner, but what can you get out of it otherwise? And I would say a few things. We're asking students to clearly communicate their scientific ideas, both their experimental design and the importance of the question that they're asking. That's a really broadly applicable skill goes way, way beyond this contest. It's a great thing to ask your students to practice. So we have Adam, who is one of our middle school winners a few years ago. And he said, this gave him his first experience in being able to write a scientific paper. That's a great question, Jackie. I love it. And then, it also, a lot of students find this contest to be really, really motivating. It gets them learning things that they never learned before. So again, this is another group of 8th graders. They, in just asking this question about problems that astronauts face in space, they started doing research, and they basically taught themselves about the field of epigenetics, which is, how we control the expression of our genes, which genes in turn become translated into proteins. And none of the students knew about this before, and it was really promoting self-directed learning, through this contest and through trying to figure out a good experiment to answer their question. And then the last is sort of more philosophical. The students are engaging in a part of the scientific process and coming up with an idea and designing an experiment. And it really, we hear from teachers every year that it's really made the students see themselves as scientists or potential scientists one day. It gives them a feel for this. And that can be really empowering for them. I'll throw in one more. It's a great thing to do this year where the students may not be in-person classes all the time. They just need to do some research and write up a proposal. None of it needs to be in a class. None of it's hands-on. So it's a great thing for the virtual classroom. It's a good assignment. And we've seen a lot of teachers using that list that way when everything shut down last spring. It was something they could give their students and they could work on their own at home. All right, so that is sort of Genes in Space 2021 in a nutshell. Does anyone have any questions? I know the process of sort of bringing that into your students can be, especially in such a busy year, can be challenging, but I'm happy to answer any questions about Genes in Space before we move on.
So do you guys have resources for research to kind of?
Yes. So we're in the process of updating our webpage for the 2021 contest cycle, but you can go there now. And there's a resources tab that has a whole host. It has lesson plans for teachers. There's a scientific design or scientific method lesson plan that you can use to implement this in your classroom. And for students, there's a number of helpful links. We've put together videos explaining PCR and gel electrophoresis using the experiment we're performing today as an example. So there's great videos to watch. There's a PCR worksheet that they can use. And it's coming this year because we're adding the additional tools. We're going to launch a webinar series that will highlight each one of the tools and give students an idea of how these tools are used on Earth and how they might be used on the International Space Station. And those, if you sign up for our newsletter at genesinspace.org, those will be announced. There'll be a schedule. There's 2 in January and 1 in February. They'll be on YouTube at one time a day, but then they'll be available afterwards. You don't have to watch them live. And there's a way for students to ask questions ahead of time that may be answered during the webinar.
Gotcha. So, question. So, do you have to use mini PCR components for all the experimentation?
So you have to design a PCR experiment for space. In the proposal, that's actually a really great question. In the proposal phase, we ask students not to worry too much about what's currently possible on the International Space Station. And that's because molecular biology resources are still fairly limited up there. If we had asked students to only do what was currently possible, they wouldn't have designed PCR experiments in the first place because there hadn't been PCR done in the International Space Station. So for that, they don't have to use necessarily mini PCR equipment. They have to use either, this year, it's either the Technique PCR, and that can include quantitative PCR, or the P51 fluorescence viewer, okay, or the BioBit cell-free system, or a combination of those. So
But you could cobble together a multi-step process, let's say ELISA or something like that.
Yeah, we'd love that.
That would be interesting in space. That might even be just by itself would be a winner? How would you pull off
Right, and so you know, like, obviously, like we want the students to be realistic. Like, don't propose sending an elephant to the International Space Station because it won't fit. But something like ELIZA, which may not have been done there before, don't worry about that at that point. If you think it should be done, you know, design the best experiment that addresses your question. Once students become finalists and winners, we kind of rein them in a little bit in terms of what's actually possible. So in the proposal phase, for example, students can propose doing these experiments on actual astronauts. In reality, it takes like 5 years to get IRB approval, and the kids will have graduated from college. So we don't do that, but they can propose it in this stage. And then we'll switch them over to a model system. I you know, again, encourage students to think about using a model system in their initial proposal, just because it's a great way to teach model systems and why they're useful. There's only 6 astronauts on the International Space Station. That's not a very big sample size, but you can send up hundreds of worms, or they have cages full of mice up there. So it's a good way of bringing in those kind of research topics, but if they want to propose a human experiment, they can. And then once, you know, as they move forward in the contest, we'll help them modify their proposals to be more practical about what's there. And if it's something like that we can get done, like the CRISPR experiment, nobody had done CRISPR in space before, but it was something we figured out how to make possible. So, you know, we do, like I said, the kids are really pushing the boundaries of what's being done up there. Other questions? And I really encourage you to sign up for the Genes in Space newsletter. We don't spam you too much. You get 1 or 2 emails a month, and you'll get all, you know, all these resources and things as they become available will be highlighted there. All right, your gels should be running pretty well by now. So I'm gonna walk you through again the experiment that we did, just to remind you, because we've been jumping back and forth between the hands-on stuff and the actual philosophy behind the experiment. And then I'm gonna ask you guys to predict the results, and then we'll go ahead and take a look and see what you guys got. So we're working with the PhilC gene. This gene has a single base pair difference in its sequence between the pathogenic E. coli and the non-pathogenic E. coli. And so we use PCR to amplify the region surrounding that single base pair difference. We made a 400 base pair fragment of DNA, and we made billions of copies of that fragment. And then today, you guys added the XMN1 restriction enzyme to those billions of copies of that 400 base pair fragment. XMN1 recognizes that single base pair difference in the pathogenic strain, and so it will cut the pathogenic strain into 2 pieces, one that's 150 base pairs in length, and the other that's 250 base pairs in length. It does not, that recognition sequence is not found in the non-pathogenic form of that gene, and so it will not be cut by XMN1. And now what you guys are doing is running those fragments out on a gel, and separating them by size. So in bands, if they're 400 base pairs, it will run more slowly through the gel and be at near the top, closer to the top, than the 2 smaller bands, which will run more quickly through the gel and be found closer to the bottom as we run it. So what I want you guys to do is take a minute to think about it. So the first 4 lanes are PCR, and we did not, those are our undigested samples. We did not add restriction enzyme to those samples. So think about how many bands you expect to see in those lanes and what size you expect. And then similarly, for the ones that we did cut with the XMN1 enzyme, P is our pathogenic control and NP is our non-pathogenic control. So how many bands do you expect to see there? So let's start with the uncut bands. And just think for a second about how many bands and what size. So A, B, P, and NP, the ones that we did not cut with restriction enzyme.
All right, so let's take a see what we got.
So does somebody want to take a guess? How many bands?
1 band.
1 band. And what size will it be?
400.
400. You got it.
0.
Does that make sense to everybody? All right. So for our PX, the one that we cut, our pathogenic control DNA that we cut with the restriction enzyme, how many bands do you expect to see in that one, and what size?
That 2?
2, and what sizes?
150 and 250?
150 and 250. And will that 150 and 250, will they appear closer to the positive electrode on your gel? So further down? Or than the 400 base pair?
Yes.
Yeah. And then how about the NP? That's the non-pathogenic controlled DNA.
1 band.
1 band. And bigger or smaller, or closer to the negative electrode or closer to the positive electrode than the PX ones?
Negative?
Yeah, higher up on the gel. That's right. And then we don't know. A and B are your space burgers and your space sushi. We don't know. Does anyone have a guess? Was it the burgers or the sushi?
I've got $100 on sushi.
$100. Uh-oh.
It's the burgers.
Cindy says burgers.
Burgers.
All right. So this is one way that your DNA, if burgers is correct, this is how your gel could look. So do you guys want to take a look? Was it the burgers? Was it the sushi? To do that, you would turn on the lights in your PCR machine, and you can either look through the hole of the folded gel, fold of view, or you can actually rest your phone camera over that hole and turn it on, and you should be able to take a really nice picture of what you see in there.
Sushi.
Dang it. I'm certain it was a sushi.
And y'all can post your pictures in the group me.
Yes, you can post your pictures. You can hold them up to the camera so I can see them.
Oh, got to get rid of the flash.
If you rest your phone on the fold of view and have the camera lined up perfectly, you shouldn't get flash. That looks lovely. So Sheena, you look like you have some primer dimer down there at the bottom, and I see your 2 bands, but it looks like your restriction enzyme. So you actually have 3 bands in some of your lanes. Does anyone know why Sheena might have 3 bands?
The loading time?
So you have 400, 100, 150, and 250. You have all 3 bands in your B and your pathogenic control lane. So any guesses why you would see both uncut and cut DNA?
Oh, because it didn't cut it. It missed it?
So that is one explanation, that your restriction enzyme might not have fully cut your DNA in the amount of time that we gave it, and that happens. There's a biological, and I'll tell you, this experiment, we've created the samples, so the biological experiment result is not relevant, but it just from a biological perspective, what might have given you both cut and uncut? Like say your restriction enzyme fully cut the sequences that it was able to cut.
Contamination?
Contamination's a good guess. So non-pathogenic E. coli is everywhere. So what I would actually expect in a burger is to see both. And so your non-pathogenic E. coli would not get cut, and your pathogenic E. coli sequence would get cut. So if you have both there, you may get 3 bands. We artificially set up this experiment. We didn't actually send you burger DNA. So it shouldn't come out that way. And what likely happened is that your restriction enzyme didn't work completely in the time that we gave it. And that may be because the full mill didn't get mixed with the sample. But Sheena, can you tell the difference between your B and your A sample, and your P and your N? Yeah. Some of them have 3 bands and some only have 1. And that is enough to tell you that it was the...
Go back to the other slide. I can't remember the lane.
So B is burgers and A is sushi. It's the burgers.
Oh, so it's the burgers.
The burgers. Does anybody else have results they want to share?
Well, mine's showing sushi. I don't care what y'all say.
And nobody can verify it because we can't all see your gel, right? Conveniently forget to post your picture.
Yeah, I don't know what happened.
Yeah.
Okay, let me see.
All right. Megan, correct me if I'm wrong, but they're supposed to save their gels for next week. Is that correct?
So if we had made a 1% gel, then yes. So no. The only thing they need to save is the buffer.
Is the buffer. Do they have supplies to pour a gel next week then?
They will.
They will. All right. Because the restriction, oh no, it isn't. Yeah, okay. So ignore this slide.
So just save the buffer for next week. And
Save the buffer for next week.
All right, so you're sending out more gel?
Yes.
Could I get another DNA ladder?
I will, yes.
'Cause I don't have one. I've used it all.
I don't have one either.
Okay.
Yeah, I've used mine too.
I mean, I'll centrifuge what I have left. I put it back to bring it back to my room, but I don't think that it'll even be enough to get 5 microliters.
Well, let me know when you centrifuge it tomorrow.
Okay, I can do that.
Okay, thanks.
Other questions about next week that Megan can answer, or maybe me.
Thank you. This is fun. I like this one a lot.
Oh, good. I'm so glad to hear that. There's something really satisfying about running a gel.
Yeah. As an add-on, I guess, since you guys get to do your implementation round, if you want to do the food safety lab to get them, interested in doing Genes in Space, that's an option.
It's a great way to kick it off. And like I said, we have a full lesson plan on getting them started on their Genes in Space proposal and some other things. And we're going to actually, another resource that we're going to be putting out this winter is converting that lesson plan to a virtual lesson plan. So if you guys aren't fully in person, we will have suggestions. We have a number of former teachers on our staff that will think through that and for you guys, try and save you a little work.
Thank you.
Yeah, my pleasure. So this is usgenesinspace.org. You can find us on Twitter, Facebook, and Instagram if you do those things and follow us, or you can join our newsletter. If you just go to the homepage and scroll to the bottom, you can sign up, and you'll get all the updates on these resources that I've mentioned and everything to do with the contest. You can find out whether or not our experiment this week that we're performing this week on the station worked, and all kinds of other fun stuff. And we have a few minutes left, which I don't know about you, and an evening is sometimes a good thing to have a few more minutes in your life, but I am happy to stay and answer any questions you have about mini PCR.
Real quick, how long do the blue gels, does it run?
If you can see your results now, you can turn it off. You don't need to run it anymore, or you can run it as long as you enjoy doing that.
Oh, okay, so there's not an automatic timer on it.
Not on that one. We have a more a newer model called the Gelato that has different voltages and an automatic timer on that one. But for the blue gel, it's our classroom model, and we figured the kids don't necessarily need that stuff.
Got it.
Yeah. So if you take the top off, yeah.
How do you fold this back down?
You should be able to press down on the seam at the top. There you go. It'll never fold as flat as when the first time you opened it, but
I don't even remember how it folded.
That looks right to me.
Yeah, good enough.
If you tuck it in there in the bag with your blue gel, it should cover it.
And for cleanup,
you can throw your gel away because you're not using it next week, and you can throw the used buffer away, but save the buffer you did not use for next week. And then all of the samples we ran, there's not enough left to do anything with, and you can go ahead and throw those away as well. In the regular trash, the stain's considered to be safe. And you'll want to rinse the buffer chamber and the casting tray and the orange cover. And the piece, basically everything but the blue base, which is not technically waterproof, you want to give it a rinse in some tap water.
So for next week, the master mix and the ladder there, we should put this back in the freezer, or is it okay to leave it in the refrigerator?
Go ahead and put it in the freezer.
Okay.
It'll thaw out quickly.
Yeah, we usually store the master mix in the freezer.
All right.
All right. If nobody
Thanks, guys.
has any other questions, then thank you guys so much for joining me. I am happy.
All
It's
these
hard to put
discoveries.
it is hard to put it in backwards. I'm impressed that you did it because it's only supposed to fit in the buffer chamber one way. I know you can make it fit the other way, but it should fit really smoothly in the buffer chamber. Putting a gel in the machine backwards is something that I did a lot in grad school by accident when I wasn't paying attention, so it is a pretty normal mistake, even for people who supposedly run a lot of gels in their lives.
And soon we'll find out if you can pour it without the clear container.
Yeah, it worked just fine. I mean,
It's not the best thing to do to your blue gel, but
probably not. But there is, you know what? I mean, it'll run. But you're right. It was moving around, and I knew something wasn't quite right.
Yeah, the clear buffer chamber is an important part of the blue gel.
You're right.
So Megan mentioned this a little bit earlier about your implementation, and I definitely, we will be able to finish this at least. It's early, but after we finish this 6 week of sessions, you will be able to choose one of these labs to implement with your class. And so whatever course you choose, if you teach one section of that or you teach 4 sections, we'll give you enough supplies to be able to implement your lab of choice with all of the sections that you teach. So as you're learning each of these and you think about where it might fit in well to the curriculum that you already have, and not as something additional as the idea. The idea is are you already teaching this concept in another way? And maybe one of these labs would be a better, more efficient and impactful, rich way to teach the concepts. So as you go through these, think about where would be a good place to kind of slide this into your existing curriculum, and that will be able to get you those supplies. So if you think something's going to be coming up really soon, like right in January when you come back from the winter break or something like that, just shoot an email over to us so that we know we can get that stuff on order. But otherwise, that is what we'll be doing on the 14th, which is the final day of all this training, is we'll be just looking at the curriculum and deciding where it can fit in naturally, not as something extra, and working through creating that whole lesson that may, be over a few class periods, just depends on your schedule. So just wanted to bring that up so it's in your mind and not kind of catching you off guard.
And the instructor's guides for each of these labs are available to download for free on our website. And we have instruction or suggestions in the instructor guide of places they may fit in your lesson plan if you wanted to check those out ahead of time.
They have
so you can,
the entire instructor guide in their notebook. So
awesome. So they don't even need to download it from our website.
Well, I think that's it then for tonight. So if you want to, I don't know if you're talking to us, Cynthia, or but you're muted. I just saw your mouth moving.
Oh, I'm talking to myself. I'm reading. Sorry, I do that. My bad.
That's myself. But y'all are free to go, and also we'll stay on for a few more minutes in case you have any questions. But thank you.
Thank you guys for joining. You guys did awesome. I wasn't sure we'd get through all of that hands-on stuff in 2 hours. You guys did a really, really great job.
Good job.
Thanks, Emily.
Thank you.
Yeah, thank you. Everything was well mapped out. It was easy for me to follow.
I'm glad. Thanks for sticking with me then.
Thanks so much, Emily. It was great.
It's my pleasure. This was really fun. I've never I've done this workshop a lot, but it's always been in person, and it's really different doing it when you're not in the room with other people.
Yeah, no kidding. Well, it went really well, I think. And I mean, this is our first time doing the it virtually, but live and hands-on as well. So we're figuring, so you're doing some troubleshooting, the whole time, but
yeah.
Fortunately, we have amazing participants.
They were great. They were willing to give it a try.
Yeah. And I really should, and Megan's been phenomenal too with all of her organization. So
yeah, I can't imagine distributing all of this. Good job.
Thanks. I had help from Janet's student, but then now we're trying to interpret some of his writing.
Oh.
So we've learned lessons about labeling things for next time.
Yeah, it's tough. Yeah, it's really tough. I know I've been putting together demo kits of some of our new labs, and I'm labeling them in my handwriting going, oh God, I hope the teacher can read this.
Well, one of the things was too, I guess, when it froze and thawed and refroze,
it wears off.
Chipped off too.
Yeah.
So we were trying to think of some alternatives to avoid that.
Basically redundancy.
Redundancy is good. We also printed, if you can print labels on your printer.
Teeny tiny labels.
They sell them. I don't know how expensive they are. Those seem to hold up better. I mean, that's how we send our kits normally.
Yeah.
But redundancy, we also use if you have, it looked like you had the colored microtubes, you could color code them.
Yeah, they did not get color coded properly.
They were rainbowy.
I think our student just grabbed whatever.
Yeah.
So nobody's matches.
That makes sense.
What about electrical tape too that might stick on there and not come off and color code that way?
Or lab tape or something.
Yeah. We'll do a couple different things.
This is great though. I'm so impressed.
Awesome. Yay.
Well, thank you for joining and teaching, and it was awesome, and we're thrilled to be partnering with you guys too. So
it's great for us. We love our connections in Georgia, and we're excited about the Genes and Space workshop coming up in January too.
Yes.
Yeah.
Excellent.
Be a group of Metro teachers. So.
Oh, cool. Yeah. All right. Well, it looks like everybody's left, or not quite left the Zoom room, but left. So
working on it.
let you guys go into your evenings.
All right. Have a good one. Thanks.
Thank you. Bye.
Bye-bye.
Bye.